Okay. Hi, everyone. It's about time, I think, we start. People are still joining, but they'll catch the main event anyway. So let's go ahead. So as it says there will be a discussion section later on, but please do keep your microphones turned off until it's your turn. And we are recording this webinar, and we'll be uploading it to the news section of our website afterwards, as usual. So our program for today, I'll start by giving a short project status overview, but our main speaker today is going to be Mira Kajanus, and Mira is a researcher at the University of Jyväskylä in Finland. She is actually running a separate project where she is working on applying Lifeplan sampling methods to the Finnish National Forest Inventory. But here in Lifeplan, she's working on the reporting of results as it overlaps with her own project. But Mira has worked up a draft report with some sample data or sample results, which she will present. And then after Mira's part, we have the discussion, for which we will have about 30 minutes, I hope. And that's where we want to hear from you about what you thought about the sample report, what you think we should add to it, something should be removed, and how you want to get your results. And if we have time, we can also go into how you get your own raw data. And then at the end, traditionally, we present the award for the most helpful team. So that'll be the 5 minutes at the end of today's session. So just to give the overview of where we are with the project. So this slide shows us how much data has been collected in Lifeplan so far. On the x-axis, we have time broken up into sampling weeks. We've got the years separated by little bars, but there's 2021, 2022, 2023 on the x-axis. On the y-axis, we have all the sites, and they are sorted by latitude. So we've got the north at the top and the south at the bottom. And the color, the intensity of the color tells you how many sampling methods were active in that particular week. So the darker it gets, and the darkest squares are where soil sampling was done as well. So you'll see the soil sampling events here in the darkest squares. Some things you can see here. Here is the block of our own Nordic sampling in Sweden, and this big blob towards the bottom is our own sampling in Madagascar. So those stay in one place; they don't do the natural urban switching. But in between them and above and below, we have the global sampling sites where the sort of sparseness is because sometimes it's the Nordic, the natural that's active, and sometimes it's the urban that's active. And you can see that we're starting to have some very nice time series building up already. Another thing you can see is that there is disruption in the sampling because sometimes equipment breaks down. So some of these lines are a bit broken, especially as you get towards the tropics in the sort of middle part. It's more difficult to keep the equipment working in the tropics. It's a more challenging environment. On the other hand, at the extreme latitudes up north, the sampling season is very short. So at the very north of Greenland, it's only a few weeks in the middle of summer. You can't read all the site names here on the y-axis, but I'll try to upload a version of this on the website that you can zoom in on more tightly so you can actually find yourself and see how you're doing. This is the Lifeplan sample pipeline. This is also on our website, and we keep it updated. So these little pictures of the sample bottles and vials, they move along as we go forward in the pipeline. So you can see that the Malay samples are the most advanced. So here, a lot of sequencing has already been done, and they've advanced to the bioinformatics stage. We have gotten initial bioinformatics from Guelph. We're currently studying those results to see what we think of them and whether we can actually improve on them ourselves on the species identifications. The cyclone samples are now firmly in the sequencing stage. Because we cut those 2 corners from sampling design, our in-house team has more time to clean cyclone samples and get them moved on into sequencing. So that started advancing much more quickly now. For camera and audio, we've been doing you have been doing the annotation, and we're building the models that will then analyze the full mass of the data eventually. But the point is that from when we get the samples to when you get species list, there are several steps that we have to go through, which is why you don't have results yet. For instance, Malay samples, which are probably the most advanced, here's a breakdown where each team's, the size of the pie tells you how many samples have been collected in total, and they're broken down by color. So the sort of red-orange is that we haven't received them from you yet. The yellow ones are ones that you have already sent. They're in Canada, but they haven't been sequenced yet. They're in the queue, and the green ones have been sequenced. And even if a sample has been sequenced, we might not have the data just yet because the data files are very large, and to download them securely from Canada takes a little time, but we are getting those. But overall, what I would like to bring to be the take-home message from this is that we don't have species lists for you yet, but when we do, how do you want to get them? What do you want them to look like? And that's what we're going to be talking about today. So I'm going to stop my share and hand this over to Mira. And there's Mira. Take it away.
Yes, thank you, Bess, for the introduction. So my name is Mira, and I'm here in Jyväskylä in Finland, and I'm going to share my screen and show some slides of just kind of like pilot results of what I've been planning to use as and present to you as some results from the data that we get. And these are all just ideas that I have come up with and how I was thinking that they could be presented. But the idea of this is that I'll shortly go through these, and I would like to hear from you how you want to see the results and what you are interested to find out from the results. So as we go, maybe like do some notes for yourself if you come up with some ideas that you don't want to have something or you want something in addition to here, so we can discuss them later. Okay, so first in the report, there'll be a general information section. It will show which team and when the report was created, and just a general status update of Lifeplan, which will be the same for all of the teams. Additionally, it will describe shortly the structure of the report, that we have, which different results of different equipment data we have, and also some terms that will be used in the report. For example, today I already used this operational taxonomic unit, OTU, which is used when we identify the DNA samples. So it's relevant for the Malays, trap, cyclone, and soil sampling. And it means that the organisms, they might not be identified to species level, so they are classified into clusters according to their similarity by the DNA markers. And these OTUs are then used kind of as a proxy for species or at different taxonomic level, for example, orders of taxonomic level we use. So this OTU is a term that you can already see today. Then we go for the DNA data for now. So all the DNA data can be presented with similar tables and similar figures, except that in Malays and cyclone samples, we only have data from the middle point of sampling. whereas the soil samples we have from 3 points of sampling, previously from actually 5, but I think now it should be from now on only 3, so the middle and 2 corner points. So in that case, they will be a bit differently presented, but just to show you the different points separately. So here we refer to the species as OTUs for each sample. We use all different taxonomic levels. However, not all of these OTUs are identified. So some of them are named as pseudo order, for example. So they are separated from each other, but we might not identify find the actual name for that order or species. Also, these results may be presented to different taxonomic levels. For example, in the fungal samples from soil and cyclones, it might be more relevant to use phylum or class level of taxonomy. Whereas with insects, the level of order is good to use because it's not as diverse, so it's easier to present the results. So the base here is that we don't want to have too many groups of the taxonomic level, so the results actually don't get too messy, but they are easy to still visualize. And in total, there will be descriptive statistics as text and tables and figures. So first, each of these different, so I'll show you the Malay sample example as an example here now, and these data are from Lifeplan Uppsala UA1 data in 2021. So there will be a text part where we can put some general statistics. Here, for example, I have now the total number of samples during that time period, the total number of OTUs that were observed, and also what were the minimum and maximum number of OTUs per sample within that time. But here we can use any kind of something that you're interested to hear. Some tables examples, one could be that we have the most prevalent here orders, as it's the invertebrates. So prevalent meaning that they were observed at the most samples from this sampling period. So on the second column, you can see how many of these OTUs were there. And the third column shows that the percentage that these OTUs represent from the entire sample that there were. Another potential table is how much were actually identified of these OTUs and to different taxonomic levels. So only less than half of them we could identify to species level, meaning that there is actually a name for them. So these are the percentages of all the OTUs that were observed, but to what percentage of those we actually have an identification, meaning that we have a name for them. However, on family level, nearly all of them already are identified. Then for the fungal samples, there's an example, as I mentioned, you get it from different points rather than only from the center of the middle point. So we can do a comparison of different points and that number of them. Here you can see only same numbers because I only had one sample from a different data set. But this is something that we could present, so you can compare the number of different phylum, for example, that were observed at different points. Then there's a list of the 10 most prevalent OTUs. For example, here you can see a Malay strap invertebrates. So here you can see all the different taxonomic levels that they were identified at and how the OTUs are actually named. So for example, here at the species level, only 3 of them have been identified to certain species types. And a second example from a similar table for the fungal samples, so the most abundant OTUs. And here you can see these pseudo species, for example, in the species column. So they have been clustered differently according to their DNA from the other species, but there's no name for these species in the database that we use as the reference data. And I've been thinking that 10 most prevalent groups would be enough here to show them. Then some figure examples. First, you can see here how many OTUs were observed in Malay samples during a sampling period. So the sampling period, which is 1 year on the x-axis, you can see here how the number of different OTUs varies over the seasons. And at the same figure, but now these OTU richness, meaning the number of OTUs in the sample, has been separated on the order level. So you can see which order, for example, here the Diptera is seems to be the most abundant, and also it seems to be the most abundant throughout the whole season. But here you could see if there's some variation over between the seasons, between the different orders. Then I'd like to add in the report an interactive Corona wheel, which you could then, there's a link for this Corona wheel that shows the different taxonomical levels here and different amounts that have been identified in the sample. And then you could click the link and go and look into this for specific order, for example, more closely in this interactive Corona wheel. I don't have an example, unfortunately, from any of our data for this yet. So this has been taken from a previous paper by Abrego et al. But here, this could be so that if you're interested for some specific order or group or at taxonomic level, you could look into them more. Then shortly about the audio and image data. So they will be presented similarly in the results as well. Like all the DNA data were clumped kind of similar output, then audio and image data would be clumped similarly as well. So these data we have from 3 different sampling points, the 2 corners and 1 middle. So we can, or whether it's 4 corners or 2 corners, depending on the year of the sample. However, we can present these results for each sampling point specifically on most data. And as I mentioned, the presentation of these results now, I have example from the bird audio data, but also the image sampling from the camera traps and the bat audio data recordings would be presented similarly. And again, we have some descriptive statistics in the form of text and then tables and figures. So here I have an example data from Finnish site 371 in Lifeplan 2021, and this is in the northern Finland, northeast Finland. So first, in the text, it explains some basis of this data. For example, that here we use data where the species have been identified with the 90% probability at least, and all the other data we have excluded so far. It also gives you some statistics how much were recorded. So for example, all these recordings have been divided into 1 minute long audio recordings, and the total of these were over 40,000, and this adds up into over 1 month of recordings. And from these recordings, 40 bird species were identified during this sampling period. Another table that we could present is how many recordings between different sites were, between different points there were. So there's one here. Here, this data has 1 middle point and 4 corner points. That's why there's five points in total. So we can see that how the recordings are divided between the different points, and also then the species numbers if they differ between the points. And we could, for each point, again show, for example, the 10 most abundant species. So this and the number of observations from these recordings that we have observed those species. So you can see some comparisons if there's different species between these points. Here's an example only of 3 of those, but it would be total of 5 here, for example. Then we have a general data in figures of the recordings. So here you can see on the y-axis the number of recordings, and on the x-axis you can see during the time of day, so how it varies with the hour of the day. So we want to see how it's presented here, that it's even, so the recordings are evenly distributed over the time of day. And the same figure for the day of the year, so how many recordings have been done over the different days of the year whether there's a variation in the amount of these recordings. And then we have a similar figure for the time of day, but now we have only recordings that have at least one species observed. So again, we can see that there's an even distribution over the time of day that species were observed, at least one. And the same figure for the day of year. And here, for example, you can see the seasonal variation that over the summertime, during the 200th day of the year, you can see a bit more recordings where a species were observed, which at least one species were observed, which is quite reasonable, for example, considering the Finnish summertime. Then we could present, for example, 3 to 5 most abundant species in your site, and we could give those species the same kind of distributions for the recordings, but species specifically now. So on the left figure, you can see how, for example, this brambling now, how the observations that it has been observed, how it divides by the time of day. And on the right-hand side, you can see how these observations are divided or distributed over the day of the year. And for example, this indicates that it's a migrating bird because it's only been observed during the summertime in Finland. So this we could, it would be easy to do, for example, the 5 species, but I wouldn't maybe go above that because then the list gets too, or the figures get too much. Then I would like to just show that we could add some such figures in the report as well, where we can see that when we have already processed the samples a bit further. So we have already fitted GLM model in the data, and then we do a prediction for the observations. So on the x-axis here, you can see the mean number of species that were observed, and the black circles represent the data, the actual true mean observations for each minute. And these have been now between 10 minutes, 1 minute recordings. So there's a mean number for these 1 minute recordings. And then the red fitted line is the predicted values that would be observed from the model, predicted by the model. So this is a step further than from the raw data. So these are some figures we could also add in there. And on the right, so on the left-hand side, you can see again over the time of the day, and on the right-hand side, you can see over the time of the year, or day of the year, how these go. Then just a short conclusion of all these that I went. So in the table so far, I have ideas to show the most prevalent or abundant species, for example, the 10 of these. Also the OTU or species richness, and this can be done at different taxonomical levels depending on the data. And also the amount of identified or unknown taxa within the DNA data. The lists that we can produce, in addition to these tables, is lists of all observed species from these different methods. So in this report, we, for example, only show the 10 most abundant species, but we are planning to also add separate documents where we have lists that lists all the species that were observed, for example, bird audio recordings. And with these lists, we can add some information for each species. For example, DNA data, the number of the DNA reads, for all the data, the number of samples that the species occur, and also the total number of observations that were observed in the audio and image data. So you can get full lists also of all the data, although this report does not include all the species. And then for the figures that I showed, we again have the OTU species richness, can be done at different taxonomic levels and between the different sampling points, depending on the data. And then also some temporal distribution over the daily and seasonal time frame, and also temporal distribution of most abundant species. For this was the species-specific figures, and also the chronoveals, where you can more interactively have a look at the data, and also then these model fits that have been fitted already to the observed species richness in the data. Some other ideas that I have had, but I haven't yet executed, is that we'd like to see the difference in the community composition over temporal range. So how beta diversity goes in or changes in temporal gradient, for example, between the seasons. So we could have a figure where you can see whether the species are much different between the different seasons. But the main topic of this rest of the time, I hope to hear and learn what you would actually like to know and what kind of details or figures or statistics you would like to know from this data that we can infer, and then I can implement those into the report and see how it would look after that. So thank you. I'm hoping some discussion from now on.
Thank you, Mira. So if you'd like to say something, there's a reactions button at the bottom of your screen, and there's the raise hand option. You can use that to ask to speak. You can also type in the chat. You can read questions and suggestions from there. So the floor is yours. And we have first, so Brigitte Gottsberger from Vienna. Go ahead.
Mm-hmm. So, hi there. Thank you for the presentation. I liked it very much. I've just got a question concerning, you said the reference database. So is this, when you look at the DNA data, is this done with BOLT or NCBI? And then also got a question about these OTUs. Is this calculated through the spins from BOLT or some other species delimitations operational things? That's my question. Thank you.
Thank you for your question. Yes, so for now, the examples I had for the Malay strap, there was BOLT database that was used as the reference database for there. And then for the fungal species, I think here it was Unite that we used. I think Brendan is here and he can maybe describe a bit closer how we calculate these clusters. So we do use those reference databases, but we also have in our pipeline some more specific calculations and clustering of these different OTUs. So they are not actually, if I have understood correctly, they're not straight referred these OTUs from these, but there's also some in our pipeline that we have developed a bit further. But does Brendan maybe have something to add to this if he's here?
Yeah, so what I can say is that the data can be made available, both treated in sort of the standardized way that BOLT treats it, so that then they would be divided into bins. But we have actually developed new statistical methods in Lifeplan and previous projects where we hope that we can get closer to the real species rather than just trusting thresholds. And so then the data that we use to do analysis within Lifeplan will use these new methods, and I think that we can probably make those tables available both in the sort of conventional bins, so that's bins for the insects, and then species hypothesis for the fungi from the Unite database, and also then our own delimitation, which we think comes closer to biological species.
Thanks,
But
Brendan.
sorry, again, just for the calculations, you would make it open how you calculated it, because in BOLT, the bins, it's not really known how exactly it's calculated. So I just would like to know if this would be kind of also published somehow.
Yes, but both the sort of description of how the method works, but also the actual software to do it will both be freely published.
Okay, that's great. Thank you.
Do we have any more questions? Katalina Ulloa, go ahead.
Hello, everyone. Thank you for explaining how the results going to be presented. It was very explanatory. I have a question about the audio mod recorded. And first, we've been having a lot of trouble. I'm from Ecuador. We are at the Putini Biodiversity Station in the Amazon rainforest. And especially the audio mods have been getting lots of trouble. Some weeks they work, some weeks they don't. We've been thinking of replacing them so we get more data. And I was thinking about, we have lots of diversity about birds in the area, and we have been reported declines on the bird populations for over 10 years. So I don't know if there's going to be a way that we can actually see this data in guilds or in a specific way so we can assess how the population trends are doing now, so we can compare with the data that we have in the past. Because we have focal observations and some recordings as well from in previous years.
So do you understand that you would like to see the bird species broken down by guild? So some kind of trait data?
Yes, Bess, could you repeat that? I lost a bit of connection.
Yeah, sorry, you're breaking up a bit. But so if I understood, you would like to see a breakdown by guild, so some kind of trait data associated with the species.
Yeah, if it's possible, yes.
We did talk about this with Mira a little bit. It would be very cool. The problem is that then we would need to have the trait data for bird species, I guess it's possible. Thomas, what do you think?
I'm trying to remember, because there is a global database on bird traits. Also, do you remember we used it for the Brazilian birds, for instance, to classify them? I mean, simply matching species identities to such a database and breaking them down by trait should be very easy to do if that was all added.
Yes, if the data is available for trait, then it
yeah, for birds there should no doubt be.
Yep.
Thank you, Thomas. Could I just add something else? We have a researcher that has worked with our bird species, so by any chance I could ask him as well if he had some database that we could use for this area. Is that all right with you?
Here, the aim is to make this automated. So unfortunately, we can't use area-specific databases. So anything that we do should be globally available, so we can use it for all the species everywhere. And that is a bit of a restriction for some points that we would like to show, but we can't do. So in that way, but you will get a species list. And then you will get, for example, like I showed that the number of observations that they were by point, for example. So in the middle point, you had this many observations of this species. So you can then further analyze that using the trait data, for example, your region of birds, if you have a very specific data for that, for example. So in that way, even though it wouldn't be in the final report, you would have the available data to then process it further with your trait data as well. But I do like this idea of, for example, looking at different guilds. But again, we have to remember that we have to have the similar kind of guild or wide enough guild restrictions that could be applied through the global scale of data that we have.
Understood. Thank you very much.
Thank you. Otso, did you want to come in on this question as well?
Oh yeah, I would like to just continue discussing about this topic and to kind of, I guess it's clear to everybody, but just to double check that of course, like the report that Mira was talking about is not the only kind of the end result of all of this data. It's one important piece, but not everything. So now what we are talking about is this kind of a report that is mainly aimed at you yourself, that, you know, this is a basic summary of your own data so that you can, you know, have a look, see if it makes sense, if you spot some obvious issues or find something interesting for you. But it's not really the final kind of a research product of Lifeplan. And now when we talk about the research products, we have of course a lot of ideas, like here in the you know, the coordinating team that what kind of a research we would like to do from these global data, and then those papers will happen at some point, and you all will be authors of them, of course, as agreed beforehand, because you are collecting the data. But then and at the point where we publish those research papers from these data, following the good scientific practices, we need to and we want to open the data. So then some version of the data will be available for the world, not the raw data, like not the raw audio and all of that, because it's also logistically infeasible. But some version of the data, like samples times species tables, we need to publish to follow good scientific practices, and we will publish them. But then, of course, we also hope that you can utilize this data for your own research in the way that Katalina was just describing, that you may have some specific ideas in your mind that, like in your case, you had some older data and you wanted to compare these data to that. And then of course, when the time is right, you will of course have access to all of your data that you have collected through this project, that it is your data. And in that sense, you can then do whatever analysis you like. The reason why you don't get it immediately now, like, what are the bird data that you have collected, is that it's not ready. We don't have the pipeline ready so that we could send you that, oh, here are the data in some nice format that just go on and analyze. And that's why we are now first practicing to make this overview summary report that already forces us to develop quite a lot of this methods development. But then, of course, in the end, you will be able to do exactly the kind of analysis that you would like to do with your data and compare to these previous data, as you mentioned. I see that there is a question in the chat which is about this kind of the same thing. Partially, if we want, can we get the raw data? And now, what do you mean exactly by the raw data? Do you mean the images and audio, for example?
Hi. Yes. I meant like the images or the audio files. If we can get that raw data, for example, if you only want the audio files, if we could get that, or if we only get the already processed data.
For the audio and the images, you have the backups yourselves on the external hard drive, so that's the raw data, and then once they are run through the model and the species identifications are done, then the output of that is per recording. For like 1 minute recording, it gives you the probability of every species, whether the probability that it is in that sample of audio. And I think it'll be for the image data, then it will be whether the species was detected in each image or not.
Oh, okay.
I think that's the raw data. And then for I can actually talk about the raw sequencing data as well. So what we are getting out of the lab in Canada is these FASTQ files, which are huge. So for each Malay sample, we get about 100 megabytes of data compressed, and then it's 10 times that when it's uncompressed. And that's coming in all the time. That's only useful to you if you know how to do bioinformatics, but if you do and you want to get your raw FASTQ files, we can send those to you. So the way that would work is that the team coordinator should request it by email, and then we'll send you, it'll take a little time to get it ready, but we'll send you a download link where you can download it, and the link will be active for about 1 week, and you should have a space to put it, so you'll be looking at gigabytes. You need a storage space ready for it and everything, but we can send you those.
Okay, thank you.
Yeah, it's a lot of data because we try to get about 1 million reads per sample, so that's why it's so big.
Okay.
Yeah. Before we go to Renata, Brendan, did you want to say something about this? Correct me.
I was just going to say, regarding the DNA sequences, the typical thing would then also be to put these raw reads into the European Nucleotide Archive short read database or something like that at the end. And so then they would be available for anyone to download if they want to spend enough time downloading them to get them. But so I believe that in the end, once the final paper is published, then all of the raw reads will be available to everyone. But that then if you need to get yours for some reason before, then we can talk to you through this method that Bess is talking about.
Yeah. Okay, now I think it's Renata's turn.
So here's good morning in Brazil, and I have a few questions and a few comments. I would like to see on the species list that you mentioned, and thank you so much, Mira, for your presentation. That was very, very helpful. I would like to see rare species as well, like the least detected ones. I don't know if 10, but like 5, the 5 least detected ones, so we can have rare species trends if we have, you know, a longer time series. I think that would be very helpful. Well, I work focused mainly on the acoustic data for our particular lab, but I mean, the entire Natal site will have the different demands from the coordinators. But I would love to see also a time series of the number of detections of acoustic events, and that not necessarily identified, but to use some sort of signal to noise ratio, like standard deviation distance, where you have detections that fall within that bracket of the signal to noise ratio distribution. You use that as an acoustic event, and you have that through time, if possible, and even better if you have through time and by frequency band. And it could be like a 1/3 octave frequency band or some other band that we agree that it's useful. And if possible to get, and then that would be like a huge table maybe, but the limits of those detections. So, you know, frequency limits, max, min, and start and end times. I don't know if you could also extract some sort of metric from those events, like, I don't know, dominant frequency, for example, or some sort of 90 percentile frequency band or something like that, you know, that will give us an idea of where the energy, the acoustic energy is concentrated in those events. That would be very helpful, because then we'll have sort of like a biophonic or zoophonic distribution of activity through time and frequency bands. So you can be looking at, you know, all these cool ecoacoustic hypotheses going around.
Thank you. I'll just respond shortly to this before you continue. This is something new for me, so that's a really good idea, and I have to look into definitely more into that. This is recorded, so I have all your proposals. I can get back to it. That's why I wasn't writing down anything, but really good idea, and I will definitely look into that. I didn't even think of that, and this is exactly why it's good to hear from you what you like to hear.
And it looks like Otso put his hand up to this one. You want to say something?
Yeah, that's a very interesting idea indeed, and it's not something that is kind of in our mainstream at the moment, because at the mainstream our analysis of the audio data are such that we try to identify these species, you know, the birds first and then later the bats, without really, you know, setting any thresholds or you know, thinking about what's the lower or higher frequency, because we just use the machine learning models to teach them to classify the species, you know, by training data. We don't hence go to these kind of numbers by ourselves. But then and we don't also have like a, you know, much drive to go to the soundscape direction or something like that, you know, we can, but it's not in our current thinking. That our current thinking is that let's go through the species. But I mean, I see that Panu Sommer here was here, so maybe he's the best one to comment. But like what I could visualize quite easily is maybe some plots that we could generate where, let's say, the y-axis is frequency and the x-axis is time. It could be, for example, you know, the time of the day from 0 to 24 hours. And then all of your data would be averaged, you know, over the whole year, so that we just see like what is the typical pattern over the day, so that you see like where do you have energy, what frequencies, what time of the day, and then we could do the same thing over the year, like Mira was showing for some data that the x-axis is the day of the year, and then we would see like how much energy at each frequency you would have on average in your data in that sense. So I think those kind of plots would be quite easy to do, like systematically for all data. And I see that you are nodding, so maybe you would like to see, for example, those kind of plots.
That would be excellent. Yes, and I do understand that the priority is just, you know, maybe something to be thinking on the like a future line of thought.
Yeah, and this plot that I said, I think that's not a big deal, that, you know, we can quite easily write a script that scans through the data and generates these kind of plots. But if you want to go much deeper, then it's maybe a different story.
Okay. In the chat, Luis Silva asked whether it's possible to remove non-target organisms such as Wolbachia from the Malaise trap summary report. I think that is possible. I don't know if we intend to do that. But Thomas, take it away.
Maybe Thomas, yeah.
Yeah, I was going to say that whether Wolbachia is a target organism or not, I think that that depends a little bit on the target person, because we do have some of the Lifeplan participants who are violently interested in Wolbachia, and because it turns out that the standard primers amplify Wolbachia fairly well. I mean, I understand Wolbachia may be an example that you picked, but because of this diversity of interests, I think our prime choice is to keep the most frequently amplified things in there, simply because they may interest someone.
And if I may add here, the report only shows, for example, the 10 most observed, but you will still get the full list. So when you look at that, you can look at the 10 most observed without Wolbachia. So you can take it kind of yourself out of there. So in that way, I don't think it's a bad thing that it's in the main report. And may I also quickly return, I think Renata was the one who suggested these rare species, to have also the list of those. And I've been thinking this, and it's really good idea, but we have to kind of, for example, with the DNA, we have to think that where is the limit that it's a truly rare species, and when it's sampled, it could be some kind of a mistake in the sampling process or sequencing process. So as soon as we get some kind of thresholds for here that we can consider true rare species, I really would like to implement this as well in there. Does also have a...
Yeah, so with the DNA data, you may well end up with 10,000 OTUs from your data. So I don't think you want to see like the 10 most common and 10 least common out of those, but rather you would like to have the access to the full list as you will be, as you will get, as Mira already explained.
Okay, Ines has a hand up from Portugal.
Hello.
Hi.
I'm sorry, my camera is broken. Well, maybe I missed something because I arrived a little bit late to the meeting, but are you going to send us this report to each site with this kind of results? Is that it? Yeah, and when it will be at the end of the project or in different phases?
Good question. So yes, the idea is to send it per each site, the report. And we were actually discussing this with Bess, that when would be the first first time. So the aim is that you would get this in periodically, not just one at the end of the project, but already during the project. We would like to produce the first one already, but as Bess earlier explained, I don't know if you missed it, Ines, but not all the data is already ready yet. So we have some data, a lot of the data we are still waiting for, and we're finishing our pipelines for the audio and images. So unfortunately, we don't have the species lists to produce the reports yet. But as soon as we have all the pipelines and processing methods for this data, we want to give you the first report. And I think what we discussed with Bess, that we would like to give you the first version of the data that we have so far is at the end of this year. So we can't promise that all the data you have sampled is ready by that time, but we would like to present to you some kind of report of the data that we already have processed and analyzed at that time. And from there on, the idea would be, for example, to give you an annual update of the report, because it's nice to plot it seasonally. So 1 year is a good way to plot it in the data. So for example, annual report could give you good information. But this is also something that we would like to hear your opinions as well, if you have some ideas. And if Bess, you have some other things to note.
No, that's exactly right, I think. We have a couple of questions in the chat. So Askar was asking about publication. So we are going to publish all at once globally the whole thing, and the co-authors will be, as agreed, max 5 per site. Ricardo Diaz Delgado will be producing separate for urban and natural, so they would be per site. Yes, and also actually already answered that one in the chat as well. Thank you. Okay, I think we probably have time for one more question if anyone has one before we go to the most helpful team part. Are there any more questions? Maybe not. Oh, Brigitte. Oh, sorry, I can't hear you. You unmuted, but we don't hear anything.
It's better now?
Yes, now we're here. Thank you.
Sorry. I'm just asking about when is it planned to somehow show or publish let's say, the whole picture, so the big picture of comparison of the different regions, and so that's just to know. Thank you.
Otso, do you want to take that one?
Yeah, so the brief answer is as soon as possible. And then the bit longer answer is that I think this, first of all, it goes separately for each data type, that, you know, we have a bit separate pipeline for audio and image and all of that. Of course, at some point we want to publish something that is based on the joint analysis of all of these data types. But if we talk about a specific data type, let's say the Malay strap data, we don't need to anymore wait for, you know, that more sampling, because we have already a great amount of sampling. So it's only how fast can we process the samples so that we are able to make the analysis that result to the paper. So Bess already showed where we are in the pipeline in terms of the, let's say, the wet lab stuff, and it's a tedious process that, you know, all of those DNA samples, they need to be sequenced. Then we need to run the bioinformatics to classify the species out of those. All of those audio and image files, they need to be classified. And to do that, we still need to develop some machine learning models and even, you know, supplement some reference databases to be able to do all of that. And we are now focusing on that part to, you know, to make sense out of the data. But we are gradually approaching this stage where we have, you know, you can conceptualize them as kind of samples times species matrices, you know, which species were found from which samples, so which sites and when. And once we have those ready, then we can move to the actual ecological questions and data analysis using, for example, the joint species distribution models that we have been developing, or any other types of analysis like descriptive analysis and whatever we would like to do. So we are, I think, equally eager as you are to get to that stage that, oh, here is the data matrix that shows which species appeared in which locations and when. Let's address exciting ecological questions based on these data. So this is the longer answer to as soon as possible. So as soon as we just get there with the quite big resources that we have, in the sense that we have something like 20-30 people working in the different aspects of these pipelines, but still quite limited resources, given that the challenges are quite big to make sense out of these kind of a global data.
I'd like to ask if you have a way to map richness by group. That would be a really cool thing to have on the report as well, like the global map of where we are, and have measures of richness for the entire data set. So we have a more, and we break it down by some sort of taxonomical level.
That could be something could be added at the beginning in the general Lifeplan summary that we have. So it's a good idea there to show that.
But now we're approaching the end of our hour, and I'm going to hand it over to Dee, who is going to present our most helpful team this year.
Hi. So if you can all see my screen.
Yeah.
So the most helpful team for this session is a team that has actually been working with us since the previous global spore sampling project. It is the team Vienna. And so they were instrumental at the start of our project because they had already set up a solar panel on their cyclone sampler and were able to give us lots and lots of advice for setting up the cyclone samplers and the batteries. And that was very helpful. They also agreed to be the first team that I could send all equipment with so that I could figure out the logistics. And so for that, we were very, very grateful. And you can also see that they have been very proactive and have fixed their own release trap. They fixed a bottle and they even fixed a tent, but I did not include that picture. So thank you very much for all your help, Team Vienna.
Thank you from me too.
Okay, may I say some words? It's Irmgard from the Team Vienna. And yesterday it was really a big surprise that we have been selected as most helpful team. We really did not expect it, and we feel very honored because we simply did what we are doing all the time. We are doing our work, and we try to help people. And for me, it's time to say a big thank you to all my team members, of course, and to all the coordination work, to the coordinators of the project. This is really a great project. So thank you, Bess and Dee, also for your work, for your constant support per email or with the Slack line and so on and so forth. We really enjoy to be part of this project, and we are very looking forward to continue this, in my eyes, really great project and successful work. So thank you again.
Thank you very much, Team Vienna. That's great. And thank you everybody for all your questions and really great suggestions. We can continue this conversation on Slack and on email. And if you have other suggestions and ideas after this, please do let us know. But for now, I think we are done for today. Thank you very much, everybody.
Thank you.
Thank you.
Thank you.
Bye-bye.
Thank you.